So welcome to the last session before the closing of this 24-hour marathon of microbiology. It is the Yeast Hour. Well, the first talk is, we'll talk about the successful outcome of the convergence of microbiology and computational sciences to provide a very successful database for those working in yeast. Well, the speaker is my colleague, Miguel Teixeira, from Instituto Superior Técnico, Universidade de Lisboa, the Institute of Bioengineering and Biosciences. Well, Miguel, please.
So let me start by sharing the screen. So can you see the presentation?
Yes.
Yeah, okay. So good evening in Portugal, good morning in Japan. It's really a privilege to be a part of this marathon of microbiology. So I really would like to start by acknowledging the invitation to be here today in this closing session on yeasts, and I'm also a big yeast fan, so it's a pleasure. So the title of my talk is Database in Microbiology, and very specifically one database, which is Yeastract. Yeastract was developed through a collaboration between our lab and a lab of computer scientists in the institute next door, the INESC ID. And it was first launched by Isabel, Sá-Correia, our hostess, while I was still a PhD student, but it's a project that we've been working on for the past nearly 20 years. So it's really very nice to be able to present this work. But before we start, I would like to go back to the 90s, and to most people, if we ask what they remember from the 90s, they would probably not remember the same thing as molecular biologists. So to see what most people would think about the 90s, I Googled it, and the first image that showed up was this one. And of course, we see a lot of familiar faces: Nelson Mandela, the Spice Girls, Jurassic Park, and a little bit of science. If you notice the right down corner, we see Dolly. But for molecular biologists, the 90s actually bring us a very nice, very important memory, which is the release of the first genome sequences. And back then in the early 90s, very demanding, laborsome, expensive projects led to the release of a couple of genomes of relatively simple organisms, such as the first bacteria in 1995, and a little bit more complex, the first eukaryotes, a yeast, of course, the yeast, Saccharomyces cerevisiae. And we think about these efforts in the early 90s, which actually helped the development of the technology that nowadays enables us to get to genome sequences fairly easy. So immediately afterwards, in the early 2000s, we were able to have access to the sequence of very important model organisms, and also the first examples of each kind. So for example, the first insect, the first plant, the first mammal, the first bird, the first fish. And nowadays, sequencing a genome is so easy that we continue to see a progression and exponential growth in the number of available genomes, as you can see in this graph. And this is due to, of course, the technological advances, which are moving really, really fast. And also, combined with that, the decrease in the cost of sequencing a genome. And just as an example, I'm showing you here what happens for the human genome. So the first human genome costed around $100 million, and at the moment the price of sequencing a human genome is below $1,000. And if we go to yeast genomes, which is the organisms that are celebrated in this section of today, the price goes down to around $300 a genome. But what hasn't accompanied the same decrease in price was actually the computation underlying the analysis of these data. And you can see here the line of Moore's law. Of course, the decrease in computation costs there is a decrease in computation costs, but they didn't accompany the decrease in the sequencing and the cost of sequencing itself. And together with genome sequencing projects, a lot of projects came by working on transcriptomics, proteomics, interactomics, and other omics, making available such a huge amount of data that definitely required professional ways to deal with this kind of amount of data. And this is still a problem today. So, of course, there were databases before the 1990s, but they had nothing to do with the databases that came afterwards. And if we think about databases in microbiology, we are deemed to bump into giant databases that go far beyond microbiology, such as GeneBank or UniProt or KEGG. But if we think about databases in yeasts, we can see a few examples on here. So, of course, SGD for Saccharomyces genome database, it's Tween database for Candida species, Candida genome database, a couple of other examples of yeast dedicated databases, some of which actually have disappeared with time. But I would like to highlight one here, which is circling around, which is the Yeastract database, which I'll be discussing right now. So if we think about first what motivated us to build such a database. So we were working on transcriptional regulation. And of course, it's easy to motivate people on transcriptional regulation if we think about transcriptional regulation as the first step of control of gene expression. Of course, if we see a gene in a genome, that's absolutely meaningless unless the gene is expressed. And how much it is expressed, so how many copies of messenger RNA or proteins are being formed from that gene, that will reflect on the phenotype. And so, of course, to study transcriptional regulation is very interesting from a fundamental point of view, but also from an applied microbiology perspective. And we were working on yeasts with two perspectives. First, yeast as cell factories. And here the idea was to be able to use transcriptional regulation data to, for example, stimulate specific biochemical pathways or stimulate fermentation stress resistance regulators so as to optimize yeast as cell factories. On the opposite side of the ring, we have yeast as pathogens. And in this case, we don't want to stimulate anything. We actually want to inhibit the activity of these pathogens. And again, it's a very nice approach to be able to inhibit transcription factors or transcription factor networks because they control on their own the expression of numerous target genes that are involved in important phenotypes such as drug resistance or biofilm formation or virulence. And so, of course, transcription regulation was an issue. And before Yeastract, what happened was every time we needed to study a gene and wanted to know which are the transcription factors that control it, or to study a transcription factor and we wished to know what the targets were, we would need to go back to reading first tens of papers, then hundreds of papers, and it was impossible to go over and over and over again the literature. So we needed something that would make this search systematic. So that's when we contacted our colleagues working in computer science, and we've decided to build the Yeastract database. It took us a couple of years to finally release it in 2006, and it was based, it is based in a very simple view of transcriptional regulation, in which we have a protein, a transcription factor that recognizes a consensus sequence, which by binding to this consensus sequence in the upstream region of a gene is going to activate or repress the expression of this target gene. So what we did was, well, look for the transcription factors, the recognized consensus, and the target genes in the literature. And together with sequence data that we got from a number of different platforms and reading lots and lots of papers, we actually put together the Yeastract database, first focusing on Saccharomyces cerevisiae. And what we've done so far was to put together, or to gather from over 1,500 papers, more than 175,000 documented regulatory associations, 310 DNA binding sites, corresponding to 183 transcription factors. So a lot of information that is now compiled in a single database. And more than a repository, we wanted to make the database accessible and also usable to people working in yeast biology. So besides putting the data together, we wanted to develop or to design queries that would enable us to look at the data in the most profitable way. And so let me give you a couple of examples. So for example, the first thing we may want to ask is how can we, or what are the regulators of a given gene? And we can do that, and let me show 2 ways we can see the response to this question. In the first case, we're seeing the potential regulators of a given gene, in this case CDR1, and by potential I mean all the transcription factors that have recognition sequences in the promoter region of the gene we are studying. Undocumented, we have all the transcription factors that regulate, that are known to regulate it, demonstrate to regulate the target gene we are looking for. As a second example, we can also identify all the potential or documented targets of a given transcription factor. And there's several ways of looking at the resulting data. There's exhaustive tables, but also a couple of more graphical depictions. And this is an example which shows that besides having an idea of how many targets, we can also filter the targets based on what's the evidence, the underlying techniques that were used to identify these regulatory associations. A third example is how we can use the Yeastract database and the data therein to analyze transcriptomics data. And for example, if you have a set of genes whose expression is upregulated in a given condition, what you can do is pick up that list of genes, paste it into this box, check for all transcription factors, press search, and what you get is something like this. And in this case, the answer of the database is YAP1 regulates 100% of your gene list. So it's, if you didn't know that already, you would now suspect that probably the transcriptional response you're observing at the transcriptome-wide level is most of it being controlled by a single transcription factor, but not just by one. It's a number of transcription factors that's working together. And I'm highlighting in this data set, what the authors that published this data set did was to identify these 4 transcription factors highlighted in red here as the regulators of this number of target genes. But by looking at the information in Yeastract, we can see that 10 other transcription factors should also be considered as regulators of this data set and may play a role in this transcriptional response as well. More recently, and that is since 2016, we've started expanding Yeastract to other yeast species. So first to pathogenic yeasts, so including 4 candidate species, and more recently to non-conventional yeasts of biotechnological relevance. And you can see the names here: Z. bailey, Cloveromyces marxianus and lactis, Commegatella paffi, and Aerophila lipolytica. And we now have in Yeastract 10 yeast species with all available regulatory data, and to this combination of sub-databases we call Yeastract Plus. And not only Yeastract Plus is a plus because it has more species, but it's also a plus because it enables us to look at these regulatory associations in a cross-species manner. And how is that helpful? So for example, you can try to predict regulation in one species based on what is known on regulatory associations in another species. So for example, here I'm showing you that the QDR2 gene in Candida glabrata is regulated by the PDR1 transcription factor. That's all we know. But we can look for who could be the regulators of QDR2, considering what is known for the Candida albicans QDR2. And what you see is this. And consider the regulators of the Saccharomyces cerevisiae QDR2, and what you see is this. So you can use this information, cross-species information, to predict a lot about transcription regulation in less well-studied organisms. And all yeasts are less well-studied when compared to Saccharomyces cerevisiae, so this can be really, really powerful. It can also help us to compare known regulatory associations. So for example, here we're seeing that for the same target genes in 3 different yeast species, how the regulatory networks are different from one another. And we can see this from a comparative perspective, but also from an evolutionary perspective, because we can eventually evaluate whether or not parts of the network are conserved, whereas other parts of the networks are not conserved. And we can also see this from a promoter region perspective. For example, here you can see orthologous genes in different species. So these are the same gene in different species. And we can see that in some cases, we have the same transcription factor binding sites in the promoter regions of orthologous genes in all species. But then in other cases, we have unique transcription factors that only show up in one or a couple of these species. and that may correspond to unique functions that have been acquired by these genes. So this could be also very powerful in terms of predicting how evolution has been working in these yeasts. So this almost final slide shows our users worldwide. We are always proud to show this slide, and we would like to consider that Yeastract has gone viral, and we have users all over the world, and we have the perspectives to maintain the information in the database regularly updated, to expand it to other yeast species. It's going too fast. Expand it to other yeast species, and of course, if you're a yeast biologist working on one yeast species which is not being considered currently in Yeastract and would like to expand it, we can talk about it, because we have also worked on the automation of the construction of Yeastract-like databases for other yeast species just based on the genome sequence alone. So this is something that can be done in a semi-automated way at the moment. So by the end of the presentation, I hope that when you think about the 90s and the early 2000s, besides thinking about all these famous people or famous movies, you also remember that the early 90s and early 2000s were the mark of the post-genomic era and the rise of a lot of very fascinating databases, including our own Yeastract. Let me just finish by acknowledging all the people involved. Besides myself, Isabel, Sá-Correia, and Pedro Monteiro, we have been working on this from the start, but then a lot of people who throughout the years have contributed by curating data or developing new tools, and also funding more recently by joining the European Research Infrastructure Leaks here. So thank you for your attention.
Thank you. Thank you, Miguel. Well, as a user, this work is means a lot to all of us. Okay, so we are going to leave all the discussion at the end. We have to close to midnight because it will be the 24 hours marathon, so we cannot exorbitate the time. So we will discuss all this at the end. And since we are closing, the idea was just to have or to be able to make a toast with beers resulting from yeast fermentation. And the idea was just to have a PhD student in his first year working with yeasts and exploring yeasts to produce beer with special flavors, and obviously accomplished scientist, professor from Japan, who will be indeed the last one. Okay, so Nuno Melo, the PhD student, please.
All right, thank you.
We need a toast, okay? We have to make a toast, please.
Can you hear me well enough?
Yes.
Okay. Okay, let me just share my screen then. All right, can you see it?
Yes.
Okay, great. So hello everyone. My name is Nuno. I'm a PhD student from Instituto Superior Técnico at the University of Lisbon in Portugal. In the last couple of years, I've worked a little bit and studied a lot about how yeasts can impact the flavor and aroma not only of beer, but of other alcoholic beverages such as wine. And today I'm here to briefly talk a little bit about the role that different yeasts can have to attribute different flavors and different aromas to beer specifically. So I'd like to begin this presentation with a very brief history, a very brief historical context. Now, as most of you know, beer is an ancient beverage. It's probably as old as humanity started to collect, domesticate cereals and grains. And actually the oldest known recipe dates back to 1800 before Christ, so almost 4,000 years ago. And it's written in the form of a poem to the Sumerian goddess of brewing and beer. And in this poem, the Sumerians describe not only the ingredients that they used to brew beer, but also the process itself. And what is interesting is that if we compare the poem back then to their deity of brewing, to the recipes that modern brewers follow today, we can see that over 4,000 years, the recipe hasn't changed all that much. So then, as now, people used water and mixed it with cereal grains to obtain a sugary syrup called the wort. They usually added some sort of ingredients. So in that poem, the Sumerians added honey. Nowadays, a very common ingredient are hops. And hops are actually flowers that have a bitter flavor, that attribute that characteristic bitterness to beer that actually makes many people dislike it. And they also have antimicrobial properties, meaning that they prevent the spoilage of beer. They prevent the growth of organisms that would otherwise, deteriorate the beer. And they have a very interesting story because in the early 19th century, when India was still part of the British Empire, obviously the British sent a lot of supplies to India, and one of those supplies was beer. However, they had a problem, which was that on the way, because it took a long time, beer got spoiled. They found, however, that if they added more hops to beer, then that beer was able to extend the entire trip to India and reach it in a state that was adequate for consumption. And it actually gave rise to a very popular beer style, the Indian Pale Ale, or the IPA, which I'm sure many of you know. And of course, while we're all here today, the microorganism that is crucial to beer making is yeast. It's this unicellular fungi that is able to transform the initial sugar-rich syrup into the beverage that we've all learned to love. So of course, 4,000 years ago, people had no idea that yeasts were the main cause of this fermentation. They just knew that if they added all the ingredients together and they left them alone long enough, they would eventually get beer. So this is what we call a spontaneous fermentation. Now, over time, we've had a change in this paradigm, particularly in the 19th century, due to the work of Louis Pasteur, who found that alcoholic fermentation was carried out by yeast. And in the last few centuries, we've actually learned a lot about yeast, and so much so that nowadays, most of beer produced is not produced by spontaneous fermentation, but by controlled fermentation. And this means that brewers select very carefully the yeast that they want to use, and they add that yeast, specific yeast, to the wort to then produce a beer with specific properties. And this is what we call the starter culture, the yeast that is inoculated or introduced in this controlled manner. And there are two main types of commercial beers today, the ale beers and the lager beers. Now, the ale beers are produced at a slightly higher temperature at around 20 degrees, and the lager beers are produced at about 10 degrees. And they both use starter yeast, so they are both controlled fermentations. In the case of the ales, we use Saccharomyces cerevisiae, and in the case of the lagers, we use Saccharomyces pastorianus. Now, these are different species of yeast, but as you may have noticed, they share the same first name. This means that they are closely related. They belong to the same genus. And this is not a coincidence. In fact, there are many characteristics that make species of this family, of this genus, to be more exact in science terms, quite well adapted to the brewing environment. Now, one of those is that obviously they perform alcoholic fermentation. They are able to convert the sugars that come from the cereals into alcohol and carbon dioxide, and obviously give rise to the beer that we all know. They are very resilient and have great survival skills and are resistant to several stresses, including different temperatures and including the alcohol that they themselves produce and accumulate. They are typically able to outcompete other yeasts in the brewing environment, meaning that over time, they typically rise to the top and annihilate the competition. And of course, because they have thousands of years, they've accompanied us for thousands of years in this brewing process, they have become very well adapted to the brewing conditions and acquired some domestication traits. This means that they've acquired certain characteristics, certain skills that makes them even better at their job, as you would expect from thousands of years of practice. And obviously, they produce a beer with good flavor and aroma. They wouldn't be used otherwise. Now, hopefully by now, I've shown you how important yeast is in impacting flavor and the aroma of beer. For one, this is because they produce a wide variety of compounds, of metabolites that affect its properties. Now, three of the most well-known ones are ethanol, which determine the alcohol percentage or the alcohol by volume, carbon dioxide, which determines carbonation, and glycerol, which determines body. However, there are many other different types of compounds that arguably, while present in lower concentrations, have an even higher impact in the properties, aromatic and flavor properties of beer, because they reach your nose, they reach your taste buds, and they're able to really determine the kind of profile you feel when you experience a different beverage. Now, of course, there are many different types of yeasts, and these yeasts produce many different types of compounds and different amounts of the same compounds, which in practice means that a beer that is produced with yeast A will in principle have very different characteristics from a beer produced with yeast B. And this means that we can explore this yeast diversity to produce a wide array of beers, from low alcohol to low calorie to beers with distinct flavors and aromas. And it's what attributes this high importance to different yeasts in the brewing sector, and what makes many researchers want to explore different kinds of yeasts to produce different kinds of beers. Now, I'd like to provide a couple of examples of yeasts that are already being explored for brewing and that actually have quite interesting potential. One of them is Torulaspora del Bruchi. It is a yeast that is found in many different environments, even in man-made environments, such as bread making. And it was actually the first non-conventional, the first non-Saccharomyces yeast introduced in the market as a starter, so the first one to be used commercially. And it produces a wide array of fruity and floral aromas that are very well suited for the production of certain specialty beers or certain types of beers like the IPA or Saison. And then we have an old staple of brewing, which is Brettanomyces bruxellensis, which is used mainly in specialty beers like the lambics produced in Belgium. And they are crucial to the profile of these beers because they produce these volatile phenolic compounds that attribute clove, barnyard, band-aid aromas to beer, which might seem undesirable, but are actually the soul behind that type of beer and very appreciated by many consumers worldwide. And finally, Brettanomyces bruxellensis can also be used because it consumes pretty much all the sugars present in the wort, including these dextrins, which are low molecular weight carbohydrates or sugars. And because they consume almost all of the sugars present, they can also be used to produce low calorie beer. Now, I'm sure many of you might be thinking, why explore different kinds of yeasts that we do not know very well, instead of just genetically modifying the yeast that we actually know and that have been used for thousands of years, such as Saccharomyces cerevisiae? That is a very valid point of view. The thing is, and many researchers dedicate their research to modifying well-known yeasts such as Saccharomyces cerevisiae to make it produce different kinds of beers and beers with different aromas and flavor profiles. The thing is that legislation, especially in the developed world, is quite restrictive. And not only that, but even public perception regarding genetically modified organisms is still quite negative in many countries. And so there are many barriers toward the entrance of these GMOs especially into the food and beverage markets. Now, luckily for us, there are over 1,500 yeast species available to explore and to apply, not only in brewing, but in many other alcoholic fermentations. And obviously many of these are pathogenic, many of these are not interesting or have no business whatsoever participating in alcoholic fermentations, but it's still a wondrous world to explore with limitless possibilities. And obviously I have no other way to end this presentation other than this. So have a pint, obviously always drink in moderation, but hopefully I've encouraged you to go out of your comfort zone, try different beverages with different yeasts, different ingredients, different methods. And luckily you can start right now because the next presentation is about an exotic Japanese alcoholic beverage. And so I'll leave you to it. Thank you very much.
Thank you, Nuno. And well, we are going to pass to the last speaker of this session is Dr. Hiroshi Takagi from Nara Institute of Science and Technology in Japan. Hiro, I'm very sorry for the 7 a.m. in Japan that you had to wake up extremely early. I'm sorry, but well, it's the last session, so we cannot. We are not
I think my time is.
And before giving you the word, I would like to inform those who are listening us, who are following us at the moment, that we are taking questions to the speakers. If you want, please, in the social media where you will be able to raise questions, and we will try to do the discussion, to make the discussion and to ask the speakers at the end. So, Dr. Takashi, Please
Yeah,
go ahead.
I think my time is too early, right? Because now 7:30 in Japan. So I have already announced my presentation time will start from the 7:40, another 10 minutes later. So
No,
Yeah,
no,
we seem to have arrived.
no
can I ask
problem, because we will have the discussion. It's better to discuss at the end. All the
Oh,
presentation.
I see. Okay, so can I share the screen?
Yes, you should be able to. Loading.
Okay. Can you hear me?
Yes.
Can you hear me?
Yes,
Yes.
we can hear you.
Okay, thank you. Okay, so yeah, it's too early, but I will start, okay? So my name is Hiroshi Takagi, working at the NIST, Nara Institute of Science and Technology. This is just only graduate student only, no undergraduate, okay? Which is located in historical city, Nara, Japan. So I'd like to express my special thanks to Professor Isabel, who kindly invited me as not only one of the speakers in this session, but also a last speaker in IMD 2020. So today, I'm greatly honored to give a talk entitled Enjoy Exotic Japanese Alcohol Beverage Brewed with My Yeast. So I think most of the countries, except Australia and New Zealand, are behind Japan. So I'd like to say good morning, good afternoon, good evening, depends on the nation where you are living. So my institute, NIST, Nara Institute of Science and Technology, is located in Nara, which is an inland prefecture near two big cities, Osaka and Kyoto. Its main industries are tourism, the textile, timber, and lumber. So Nara occupies an important position in the history of Japan. It is said that Japan's first state, Yamatai, was located here. So Nara is also a home of various World Heritage sites, such as Todaiji Temple and Horyuji Temple, and others, including numerous Buddhist art and architecture, classified as national treasures and important cultural asset. Also, Nara is very popular for lovely deer who are messengers of the gods. So now I'm the president of International Commission on Yeast, ICY, which is a part of the Mycology and the Eukaryote Microbiology Division of the International Union of the Microbiological Science Societies, IUMS, since back to 2016, in which I organized the ICY General Congress in Japan. At that time, I invited Isabel as one of the plenary lecture speakers. Do you remember? Yeah. So I was a research scientist of the Japanese food company, Ajinomoto, amino acid producers, for 13 years. So back to 1995, I decided to change my career to the academic position in local university in Fukui, Japan, and moved to NIST, Nara, to the 14 years ago. Now, these staffs are working with me, and we have so many graduate students. They're including those from the Southeast Asian countries. My research background is mainly applied molecular microbiology using yeast and the bacteria. That covers the basic studies in microbial science and practical application in biotechnology. So I will briefly show you once again the yeast Saccharomyces cerevisiae. So this is an important microorganism in basic science as a model for higher eukaryotes. Because the past Nobel Prize winner, Professor Yoshinori Osumi, discovered and elucidated the mechanism for autophagy, which is the fundamental processes for degrading and recycling the cellular components, proteins. Also, the yeast is a useful microbe in fermentation industry due to its extremely high ethanol productivity. So for example, I recently isolated the new yeast mutant that overproduces the isoamino acetate known as the key flavor. So by brewing with my yeast strains, the awamori, which is a traditional Japanese distilled Japanese alcohol beverage, has been successfully commercialized. So basically, I personally love the drinking any alcohol beverages. Also, my main research project is involved in the ethanol tolerance of yeast. Okay, but I am sensitivity to high ethanol concentrations of ethanol at night. Okay, so yeah, there are two types of the alcohol beverages as shown here. Non-distilled beverage and also distilled spirits. So among them, the sake and the shochu are very unique Japanese product by good collaboration with the fungus and the yeast. So sake is made from the steamed rice by multiple parallel fermentation of the fungus Aspergillus oryzae and the Saccharomyces cerevisiae, which produce the saccharification enzyme and ethanol from glucose, respectively. So recently, we successfully commercialized the new sake containing the amino acid ornithine, rich high level of ornithine, which is the physiological functions such as reducing the fatigue and improving the sleep quality. Brewed with my ornithine overproducer of sake yeast mutant. But today, I will focus on the awamori brewing. What is awamori? So one type is shochu, which awamori is a traditional distilled alcohol beverage made from the indica steamed rice in Okinawa, which is the subtropical islands in Japan. So its brewing process is quite different from that of other distilled spirits like whiskey made from the grain mash, the foods where the conversion from the starch to alcohol via sugar by malt enzyme and yeast. So during awamori brewing, the fungus Aspergillus luteinensis and yeast Saccharomyces cerevisiae are doing very good jobs for the preparing the fermented mash moromi and the producing ethanol, respectively. So after the fermentation, the alcohol is mainly distilled under atmospheric pressure and then is put in a barrel for aging that develops a rich and strong flavor. Okay, so isoamino acetate is known as the banana-like flavor and the key flavor, ginjoko in Japanese sake, in yeast. Isoamino acetate is mainly converted from the intermediate alpha-keto isocaproate, which is an intermediate of leucine synthetic pathway. So in 3 steps, they're catalyzed by alpha-keto acid decarboxylase and alcohol dehydrogenase, and finally alcohol acetyltransferase. So alpha isopropyl malate synthase, IPMS, as shown here, is the key enzyme that controls the leucine synthesis because its enzymatic activity is subjected to the feedback inhibition by leucine. So to increase the isoamino acetate contents in our moly, we try to isolate yeast mutant that accumulate leucine in the cell by a conventional mutagenesis. So I will briefly show you how to construct the commercial almond yeast. For the application of the recombinant yeast, self-cloning yeast, which contains no foreign genes or sequences except for yeast DNA, is more acceptable for consumers than the genetically modified yeast, GM yeast. According to the Japanese government guidelines, the self-cloning yeast does not have to be treated as the GM yeast because the self-cloning processes are considered to be almost the same as naturally occurring gene conversions. However, the food industry has not yet accepted self-cloning yeast. So I constructed our moly yeast strains by a conventional mutagenesis, which is a widely acceptable method of the yeast breeding for food industries and consumers. Okay, so how can we isolate the yeast cells with the leucine accumulation? It is generally said that the toxic leucine analog 555-3,4-leucine called the TFL is transported into the cells in the leucine transporters. It causes the misfolding of proteins into which it is incorporated competitively with the leucine, and thereby the inhibits cell growth or cell death. However, the leucine accumulating cells are resistant to TFL to detoxify or to dilute the TFL concentrations in the cells. So we isolated TFL-resistant mutants derived from the wild-type our moly yeast. Okay, so this slide shows the method, how to isolate the TFL-resistant mutants. So basically, the parent cells cannot grow in the presence of the high concentration of TFL due to its toxicity. However, after introduction of the random mutagenesis into the genome of many cells with the ethyl methansulfonate EMS, the several colonies were shown up on the agar plate containing a TFL. So then such TFL-resistant mutants were collected and tested for intracellular leucine level. So among the many TFL-resistant mutants, we successfully isolated a very good mutant, 18-T55, with the increased leucine content in the cell, and found the heteroalleic mutations in the LU4 genes encoding IPMS, particularly the serine 542 phenylalanine and alanine 551 variants of IPMS. The leucine content in cells expressing the IPMS variants was higher than cells expressing the wild-type enzyme. So IPMS activity of the wild-type strain was markedly inhibited by leucine. This is the wild-type enzyme. But the mutants show about 80% activity, even in the presence of high concentration of leucine. Also, the IPMS activity were insensitive to feedback inhibition compared with the wild-type enzyme, so leading to the leucine oversynthesis. So interestingly, the homology modeling of IPMS suggested that the leucine binding was drastically inhibited in these variants due to the strict hindrance in the cavity. So we then cultured the yeast strains and measured the flavor contents from the volatile substances by gas chromatography and mass spectrometry. In this mutant, the both leucine and isoamino acetate contents were higher than those to the parent strain, compared to the parent strain. Also, in proportion to the intracellular leucine level, the yeast cells expressing the IPMS variants produced higher isoamino acetate than the cells expressing the wild type IPMS. So these results indicated that the IPMS variants that with removal of feedback inhibition by leucine causes intracellular leucine accumulation and also extracellular hyperproduction of isoamino acetate. So finally, in collaboration with the Okinawa's venture company, Biojet, the awamori brewed with my yeast mutant strains 18-T55 was successfully commercialized 4 years ago by the oldest brewery in Okinawa, Shinzato Shuzo, as the brand name, the Hyper Yeast 101. We very recently isolated a novel Saccharomyces cerevisiae strain from the hibiscus flowers, hibiscus yeast, called hibiscus yeast, that produce the high level of ethanol and the 4-vinyl glycol, which is a precursor of vanillin, known as the key flavor of aged awamori. However, the isoamino acetate concentration observed in hibiscus yeast was lower than that observed in the conventional awamori yeast. So interestingly, by the whole genome sequences, we found that the hibiscus yeast is contiguous to the wine yeast and the different clades with the classical awamori yeast in the phylogenetic tree, suggesting that the hibiscus yeast does not share ancestry with the sake or shochu awamori original yeast strains. So to increase the isoamino acetate content of hibiscus yeast, we obtained the excellent mutant T25s with the leucine accumulation by conventional mutagenesis. In this mutant, we identified the heteroalleic mutations in the LUFO genes, encoding the glycine 516 serine variant of IPMS that was less sensitive to feedback inhibition by leucine and leading to the leucine intracellular leucine accumulation. In a laboratory scale, awamori brewed with the strain T25 showed a higher concentration of isoamino acetate than that brewed with the parent strain. So finally, this awamori was recently commercialized by the local brewers in Okinawa as the brand name Hana and also Sho Kamimura. I think there's such a combinational approach to yeast isolation with the whole genome analysis and metabolism-focused breeding has the potential to vary the quality of alcoholic beverages, not only awamori, but also other sake or beer and wine. Okay, so this, from now, I'm not the scientific section, but I'd like to give you many information about awamori. I'm happy to instruct you how to enjoy awamori. So many people drink awamori straight or on the rocks, water and ice, to enjoy its original taste and flavor. However, there are many ways to drink it with water. Okay, first of all, if you want to enjoy the mellow fragrance, the recommended ratio of awamori and water is 3:2 or 1:1. On the other hand, if you want to quench your thirst on hot days, a suggested ratio of awamori and water is 1:2. And also another way is to leave it in the refrigerator overnight. So doing so will mellow the taste, showing the new world to you. Okay, finally, please let me tell you how to get my awamori. Unfortunately, two kinds of awamori are currently not available officially due to they are sold out. However, there are several ways to get them. At first, directly visit the local brewery in Okinawa, Shinzato Shuzo for hyper yeast, and Kamiya Shudosho for Hanahana, which are located in the subtropical islands in Okinawa. Or purchase the Sho Kamimura at their website of Amazon.com. So now you can buy the almost €27 or 32 US dollars, okay, by Amazon.com. Also, I will encourage you to donate 1,000 US dollar or 800 euro to the our institute fund. Okay, so if you donate such kind of high money, you can get my awamori set or Japanese chopsticks. However, the most efficient way is to become a friend with me. Okay. So regarding today's topics, my former and the current staff, postdoc, and the student did a great contribution. Also, I'd like to thank all of these collaborators. Finally, these works were supported by such kind of grant from Okinawa and also Japanese government. So finally, microbial products makes us happy, including foods and beverage, the commodities, and the pharmaceuticals. I love microorganisms because they do not betray us as far as we handle fairly. Thank you very much for your listening.
Thank you. Thank you very much. Very nice talk and very adequate to close the session. Yeah. It's a pity that we do not have your awamori here just for a toast.
Yeah, I will bring the awamori
but okay,
in the next international conference.
And I would like to inform you and all our followers that this session, as well as all the 24-hour sessions, are being recorded and were recorded. So they will be available. They will be available at the International Microorganism Day and the related FAMs and associated social media. Just to tell you that it was not a problem that you start your presentation before the time, because they will be freely available and everybody will enjoy your talk and all the other talks during the day. So we have 5 minutes, as you see. It will be a very short discussion. I don't know if the three speakers would like to ask questions each other. Okay, Miguel, please.
Thank you, both of you, for your very nice talks. I'm thirsty right now, and I would like to be your friend. So, maybe a question to both of you. The two of you talked about the use or non-use of genetically modified organisms, and there's some ways to go around it, like the classical mutagenesis. Do you think there's something wrong with the law in this respect? Are we being too strict? Is it not the same to use genetically modified organisms or use classical mutagenesis? What's your thoughts on that?
Hiroshi, do you want to comment, or should I?
Yeah, you first.
Okay, so obviously, as a scientist, even if someone who's starting now, I'm a bit biased towards saying that yes, the legislation is a little bit too strict in regards to GMOs in food and beverage. At the same time, I do understand the concerns, but it's often doesn't make sense that something that is random mutagenesis would be acceptable, while something that is targeted mutation would not. For some reason, there's the dividing line in terms of public perception and even in terms of legislation that, in my opinion, it's not justified.
Yeah, probably. Yeah, today as I showed you, the hibiscus yeast, was also the mutagenized with the EMS. So, but recently, as you know well, the whole genome sequence technology are well developed. So it is not so difficult to get the whole genome sequence, not on to find out not only the genes which contribute to the favorite purposes, but also other favorable or non-favorable mutations become clear. So as one of the sales stock, we can give many information to the consumers, even though there's so many mutations in the genome of the new yeast mutant or strains. Yeah. So still, I think still the conventional mutagenesis is very powerful approach by the combination with the whole genome sequence or metabolism, metabolic mix analysis. Okay? But basically, as in Japan, also as the European country, also there's still particularly food industry and consumers do not accept the GMO and the even the self-growing yeast. But we are discussing the how safe their genome editing.
你早上太来了以后，太早来了以后太疲劳了。昨天休息好了吗？
We're about to close the session. Well, we are closing the session. At this moment, I would like to give the word to Joseph from the FEMS office. But before, I would like to thank and to congratulate FEMS and all the speakers for this perfect marathon of microbiology. It was a privilege for me, and I'm pretty sure that for all of us that were here in this platform to interact with so nice people from the office. Thank you very much, Joseph. Thanks, Ben. Thanks to all the FEMS office and the FEMS direction. And obviously, thank you all the speakers of today. And I will pass the word to Joseph